Good afternoon, everyone. I'm Weonjung Sohn, under Professor Sean and Dr. Sharaf's supervision, and I would like to talk about my research topic today, which is compact and high-performance activated carbon incorporated membrane bioreactor to transform urine to fertilizer for a circular economy. I'm going to start from the research background, including literature review, and then research objectives, and the methodology and results up to date, and finish with conclusions and my future research plans. So, as you can see in this graph, the total consumption of synthetic fertilizer has increased nearly 300% by 2019 compared to 1961. And moreover, if you have a look at this table, the global demand for nitrogen, phosphorus, and potassium for fertilizer use has increased by 16 million tons, and nitrogen demand was the highest, accounting 6.5 million tons increase. Because this continuous increase of synthetic nutrient demand can result in global energy intensity and depletion of limited resources on the Earth, there is a significant need to find a substantial nutrient provider. In fact, from the past, we have been living in a linear economy where we take resources and make and use, and then we dispose. To make a synthetic fertilizer, nitrogen is fixed from the atmosphere by Haber-Bosch process, which consumes 1 to 2% of global energy demand and also causes greenhouse gas emissions. And phosphorus, potassium are usually gained from the mining, which is predicted to be depleted in near future. Therefore, now we should transform this linear economy into a circular economy by closing this loop. And one of the most promising solutions can be recycling our urine by transforming into a fertilizer. Human urine accounts for only 1% of total wastewater volume. However, it is responsible for 80% of nitrogen, 47% of phosphorus, and 71% of potassium in total nutrient loadings for wastewater. Therefore, when the urine is source separated by these novel urine diversion toilets, it is expected to improve conventional wastewater treatment plants to be energy efficient and cost effective, because the operating energy and cost for the aeration and nutrient removals can be reduced. Everyone produces average 1.2 liters of urine per day. And urine consists of 95% of water and 2% of urea, nitrogen, and the rest of other ions. Although the composition of urine can be different depending on different ages, diets, and physical activities, it is rich in nitrogen and phosphorus, potassium, which are the major nutrient components for growing plants. And this also shows high potential for replacing around 25% of synthetic fertilizer use. The composition of fresh urine and stored urine can also be different due to the urea hydrolysis. In the fresh urine, most of nitrogen exists in the form of urea, and the pH is between 4.2 to 6.9. However, as soon as it contacts toilet walls or pipes, urea is hydrolyzed into ammonium in very short time. During this urea hydrolysis, because of alkalinity produced, pH level increases up to 9.2, and most nitrogen is in ammonia-nitrogen form. According to the ammonia-nitrogen equilibrium, when the hydrolyzed urine is acidified, all the nitrogen exists in the ammonium ions, therefore prevents ammonia volatilization. And when it is alkalized, ammonia form can dominate and can be recovered by stripping. So many previous studies have investigated various technologies to convert urine into a more convenient fertilizer to use, because direct application of urine to land has unpleasant smell and high ammonia volatilization, and also unwanted organics and micropollutants. The technologies can be divided into physical, chemical, and electrical and biological methods. Firstly, simple storage and distillation, freeze-thawing, and membrane separation process can be included in this physical treatment method. However, there are several drawbacks. For example, for storage and distillation can cause high ammonia volatilization, and distillation and freeze-thawing is energy intensive. And for membrane process, pre-treatment of urine is necessary to avoid organic membrane fouling. In case of chemical treatment, such as acidification, alkalization, precipitation, adsorption, and ammonia stripping, all the process requires a lot of chemical additions, and therefore it is expensive. And in case of precipitation of strobite and ammonia stripping, they can mostly recover one type of nutrient, either phosphorus or ammonia. Electrical treatment, including MFC, MEC, ED, and CDI, is not widely studied yet, because further stripping process is required for MFC and lower recovery rate compared to other technologies. Urine can also be applied to microalgae as a growing medium, but it is hard to apply with undiluted urine. This nitrification, which means biological ammonia oxidation, can potentially be the most promising technology which can overcome previously mentioned drawbacks. In aerobic condition, nitrification occurs by two sequential steps. Ammonia oxidizing bacteria, AOB, firstly converts ammonium to nitrite, and then followed by nitrite oxidizing bacteria, NOB, converting nitrite to nitrate. As alkalinity can be consumed during this nitrification, the pH level decreases. This process is commonly known as two-step nitrification process. However, recent study discovered the presence of complete nitrification bacteria called Comamox, which can oxidize both ammonium and nitrite as a substrate. The best technology for this biological nitrification is membrane bioreactor, so-called MBR. Firstly, MBR has high operational flexibility by separating HRT and SRT, as well as minimal sludge production due to long SRT compared to conventional activated sludge system. Also, when urine is applied as a feed to MBR, the nitrification allows a theoretical maximum 50% of ammonium conversion into nitrate, and this can lead to the pH level drop to 6.2 without any chemical addition. In addition, malodorous organics can be degraded by heterotrophic bacteria in activated sludge. Finally, nitrogen, phosphorus, and potassium can be recovered in one final product as a liquid fertilizer. Fortunately, in our UTS Building 11, we have sewage pipes which can separate urine from waterless male uriners in every floor. And this network allows collecting source-separated urine and storing in a water tank. Both lab-scale and pilot-scale MBR have been studied for urine nitrification, and the nitrified urine, which was concentrated by membrane distillation, successfully applied as a fertilizer, showing compatible results with commercial fertilizer. However, there are still some challenges remaining for this MBR system, which can be divided into 3 sections. So first one is long hydraulic retention time, HRT, of MBR, which is reported to be 10 to 20 days in the previous studies. And this long HRT is related to treatment capacity and results in large footprint considering building scale MBR. So the addition of biofilm carrier can be one of the solutions, since it can provide large surface area for slow-growing nitrifying bacteria to attach and grow stable, and the resulting more biomass for HRT reduction in small footprint. Secondly, because AOB grows faster than NOB, the intermediate product, which is nitrite, can be inevitably accumulate during the nitrification, and this high nitrite concentration can inhibit NOB. This not only results in long startup time, but also unstable operation. Therefore, COMAMOX application can mitigate this nitrite accumulation. Finally, micropollutants in urine can limit the production of safe fertilizer. Although our group already applied activated carbon addition to MBR and demonstrated high removals of several micropollutants removal, investigation of various micropollutants in real urine and their removal is not yet studied. Therefore, my research topic focuses on, firstly, for HRT reduction, I will investigate the performance of improvement of biofilm carrier supported MBR with powdered activated carbon in comparison to conventional MBR in terms of nitrification and HRT. And then, as a solution for nitrite accumulation, I will evaluate the feasibility of COMAMOX bacteria for the complete nitrification of source-separated urine and its effect on mitigating nitrite accumulation. And also, in order to remove micropollutants in real urine, I will explore the effect of PAC addition to MBR inoculated with COMAMOX on the removal of micropollutants in urine. And finally, based on the results from Objective 1 to 3, I'd like to investigate the feasibility of high performance pilot scale MBR for the nitrification of source-separated urine and transformation into a fertilizer for circular economy. So as my experimental progress up to date is focusing on Objective 1, I would like to present my the results of Objective 1. For my material preparation, source-separated urine was collected in the basement of our Building 11 and stored in a water tank for lab-scale MBR operation. And powdered activated carbon was obtained from our environmental lab and washed and dried overnight before use. The biofilm carrier used in this study was made from polyethylene with 30 millimeters diameter and 1.1 millimeters of thickness. The reason why I chose this biofilm carrier is firstly high surface area for biomass growth, as well as lightweight, so that less aeration energy compared to other types of carriers. And also because this disc and parabolic shape can have high oxygen diffusion for active biofilm formation and prevent the anoxic zone for denitrification. This biofilm carrier was added from the beginning of operation, MBR operation, to support bacterial growth. But PAC was added after the carrier's surface was saturated with biomass and started to sink down to avoid PAC accumulation on the carrier surface. So this figure is showing my schematic diagram of my lab-scale MBR experiment. The feed urine was controlled by pH-based dosing pump, which means pH level was maintained at 6.2 by dosing high pH urine whenever the pH drops in the reactor. And because of this, HRT can be varied over time. And SRT was set as infinite except for the samplings. And initial MLSS and MLVSS was around 4.5 and 3.9 respectively. And dissolved oxygen was maintained at 4.5 milligram per liter, which was enough aeration rate to fully mix both reactors. And the membrane flux was maintained at 3 LMH, and water level sensor was maintained. Water level could maintained by the water level sensor connected to another membrane. And MLSS, MLVSS was measured by standard method, and nutrients were analyzed using test kits, IC and ICP-MS, and TOC was analyzed by the TOC analyzer in our environmental lab. So the two MBRs were operated in parallel during 120 days of operation to compare between conventional MBR and MBR with biofilm carriers and PAC. The feed urine was started with 10 times diluted urine in the first week, and the dilution rate was gradually decreased. When the ammonia nitrogen concentration in the feed was increased over 3,000 milligram per liter, the nitrite concentration started to increase. Therefore, the feed nitrogen concentration was reduced to 2,000 milligram per liter until it was stabilized. And also the dosing pump was turned off to reduce nitrogen loadings. After the recovery, undiluted urine was started to fed with relatively stable operation. So overall, MBR with carriers showed 25.9% faster recovery time from nitrite accumulation, and the startup time shortened by 14 days compared to conventional MBR. And the average HRT during the time when the undiluted urine was fed was average 10 days and 6 days in the conventional MBR and MBR with carriers, respectively. And this graph is showing only nitrite concentration and conversion rate. Although the nitrite accumulation occurred in both MBRs, the ammonium to nitrate conversion rate showed more stable and higher rate due to the attached growth of slow-growing nitrifying bacteria on carriers. An MBR with the carriers showed higher nitrification rate due to the shorter HRT and higher conversion rate, which was 304.2 milligram per liter per day, while the nitrification rate in conventional MBR was only 193.8 milligram per liter per day. The initial MLSS was 4.54 gram per liter and 4.7 gram per liter in the conventional MBR and MBR with biofilm carriers, respectively. And overall, both MBRs showed the increase in MLSS as well as MLSS ratio. The conventional MBR showed the increase in MLSS concentration up to 5.55 at the end of the operation, along with the increase in the ratio from 0.82 to 0.88. On the other hand, in the carrier-added MBR, before the PAC addition, the increase in MLSS was rarely noticeable. This might be due to the attachment of suspended biomass to biofilm carriers. But after the PAC addition, it resulted in significant increase in MLSS by 0.7 gram per liter, as well as MLSS ratio from 0.84 to 0.9. The raw biofilm carriers was added from day one, and after saturated with attached biomass, they started to sink down with total 2.82 grams of attached biomass at day 59. The attached biomass at day 115 was 2.96 gram, which means that PAC did not accumulate heat on the surface due to the parabolic shape could remove the excess deposition by shear forces. PAC addition in the MBR with biofilm carriers increased the TOC removal rate by 10% from 86% to 96% on average due to the superior absorption capacity of PAC. And after PAC addition, TOC removal rate was almost 12% higher than that in conventional MBR. So overall, the major nutrients, nitrogen, phosphorus, and potassium was well retained in the MBR, achieving high nitrification, which implies that the complete and efficient nutrient recovery in one product as a liquid fertilizer with NPK ratio of 6 to 1 to 1.5 was achieved. As conclusions, the MBR with biofilm carrier addition showed faster recovery from the nitrite accumulation and shortened the startup time compared to conventional MBR. And also the MBR with biofilm carrier and PAC addition was able to reduce HRT and increase nitrification rate as well as improve TOC removal efficiency. To sum up, the MBR with biofilm carrier and PAC showed feasibility to produce 6 to 1 to 1.5 mpk liquid fertilizer in a more compact and higher performance reactor compared to the conventional MBR reactor. And this table shows my research timeline. So I finished experiments for Objective 1, and microbial analysis, such as DNA sequencing, will be also conducted to see microbial composition and difference in conventional MBR and MBR with carrier and PAC. For Objective 2, I started acclimation of Comamox bacteria, and this will gradually acclimatize to high nitrogen loading rate of urine. And I'm going to start experimental design of Comamox MBR as soon as it is enriched. After that, I will focus on the existing micropollutants in urine and their removals by PAC addition, along with long-term operation of MBR. And then, based on these experimental results, pilot-scale MBR will be operated for real application of compact and high-performance MBR to produce green fertilizer. And then I'll finish with thesis preparation and CA3 at the end of 2025. Yeah, thank you for listening.
Thank you, Weonjung. You made it exactly 26 minutes. Well done. Okay, good presentation. I will start with the assessors. I guess we'll start with Dr. Yang.
Okay, yeah, well done, Weonjung. It seems like the literature review is very nicely, thoroughly done. Although you showed a few slides, but I read the reports and it seems like it's very thorough. So it's good. I think the delivery was also very clear. So I have only a few questions, technical questions. So the first one is, so use the biomass carrier, which is made out of the polyethylene. And as far as I'm concerned, the polyethylene is not very hydrophilic polymer. So could you tell me about what's the role of this biomass carrier, and is there any room for improvement of this this material, like a surface modification or changing the morphology or things like that?
Yeah, so the major purpose of addition of this biofilm carrier is to, it's as a supporting medium for biomass growth. So basically the carrier has high surface area with porous, high porous. So the growing nitrifying bacteria is slow growing, so they can attach on the surface of the carrier, and then they can, when they attach and grow, they can stably grow then the suspended condition. So that's the major role of this carrier. And I don't think I need the surface modification because it has high porous, a lot of pores like PAC, but it's a kind of chip-shaped shape, and the major purpose is just attaching the biomass. So high porous and active area is the most important part. So I don't think the surface modification is needed.
Okay, thanks. And luckily you have access to the separated source for the urine. And but most of our toilets, it's not separated. So in that case, what's the main issue? Is it only the economical issue to separate those urea from the mixed waste, or it's theoretically impossible to separate them?
You mean in the wastewater plant, treatment plant, or in the toilet?
In the toilet.
In the
system.
toilet system.
Yes.
So we use flushing toilet. So when we flush the water, it comes down together with the water, urine. So that's not the source separation of urine. So it goes to the wastewater plant, treatment plant. But in our building, only in male uriners, they don't flush with water. So only the urine goes down to the pipes, and then we can collect and source the separate urine from water.
Yeah, so what I'm asking is if it's flushed urine, is that the matter of the the diluted concentration is only the problem, or is there any other problem causing from the mixing with the tap water?
So the reason why we use source separation of urine is to. So when it's combined with water, it's basically the same as wastewater treatment plant. And sorry, I don't know how to explain, but I mean, diluted urine is not affecting the MBR or technical part of this process, but we prefer source separated urine because we can also recover water for water.
I see.
And also separated urine can be separately treated in the process.
Okay. Right. Yeah, I think that's all I have.
Thank you, Dr. Yang. We can go on to Dr. Leonard.
All right, thank you very much, Daphne, and well done, Weonjung. I feel like your presentation was very clear, and you have prepared very good slides as well. And I also reiterate what Yong was said about the literature review is quite comprehensive. So it's well done on that. In your CA1 report is quite comprehensive. Although I found that one of the main objectives that you will be doing is about using Comamox, right? The bacteria.
Yeah.
But it's not very well discussed somewhere in your literature review. I didn't really see much of the information about that, which I feel like this is one of the major activities that you will do, right, for nitrification. So if you could possibly add more of this information and details in your literature review in the future, that would be really be helpful. So the question that I have is with regards to first probably is the addition of, it looks like you have initial results are quite good. You have reduced your HRT, and also especially with the HRT you have reduced that one, and you have probably removed some micropollutants already as a start. Did you mention that one with the addition of PAC or not yet?
In my experiment, it hasn't been removed, but in the previous, our group demonstrated that PAC addition could remove several micropollutants.
All right. My question probably is in terms of long-term operation with the addition of this biofilm, the addition of your how do you call that, the additional surface area for carriers and the PAC, how would it affect in terms of the life of the membrane that you're using? Because there's aeration and everything. I understand there's some, it will help probably in reducing fouling and everything, but it will also probably be exposed more on the membrane. How do you think it will affect in the overall lifetime of your membrane for this process with MBR plus all of these biocarrier and PAC?
Yeah, that's a good point. So actually, I was focusing on the improvement of nitrification and HRT reduction.
Yeah.
So I only focus on this part, but in the long-term operation, when the aeration aerated and then the circulates the biofilm carriers and PAC, it will probably affect membrane as well. So that's one of my maybe future objective as well, and how just to see effect of this biofilm carrier and PAC on membranes, foulings and membranes, and also after that I'll figure out how to like reduce this membrane abrasion by these carriers as well.
You mentioned in your research plan as well that you'll do some microbial analysis, right? Are you doing this by yourself, like DNA sequencing? Do you have enough background to do that, or is somebody helping you out?
Yeah. We are sending samples to for the DNA sequencing. The external, yeah. So,
Okay. So you have your background on analyzing the results as well for that, or somebody's helping you?
we're asking somebody help to analyze the result.
Okay, that's fine. Okay, I'm just curious about CommonMox. So that's your objective too, right? And so in your process, you are going to use CommonMox bacteria instead of the usual AOB and NOB, right? Is that what you are planning to do?
Yeah, I'm planning to enrich CommonMox instead of AOB and NOB from activated sludge.
Yeah.
But actually, we'd like to do the DNA sequencing and see composition of current MBR, because CommonMox might be there. So after we have the results from the DNA sequencing and the composition of this activated sludge, we can see how AOB and NOB dominance between the CommonMox and AOB and NOB. And then because CommonMox can reduce the nitrite accumulation because it can oxidize both ammonium and nitrite. So I'd like to apply this CommonMox, maybe only this CommonMox inoculum to MBR instead of AOV and NOB to reduce this nitrite accumulation.
All right. So it's a single CommonMox bacteria in the system, and then and also you're trying to see if what you have right now is a combination of AOB, NOB, and CommonMox, and what is the which bacteria is dominating in that system, right?
Yes.
Okay. Okay, that's okay. That's a good plan. You say you're working on that one right now. Your ongoing enrichment.
Yes.
In terms of micropollutants, so you are using PAC to remove micropollutants. I think I also probably heard somebody also doing the same thing before, right? So you're just combining, yeah? There was another student probably using PAC to remove micropollutants.
Yeah, Jade is.
Oh, was it? Okay. So now in your case, if you are trying to combine these things together with CommonMox, if CommonMox is going to work. Okay. Which micropollutants are you looking at? Are all kinds of micropollutants in urine?
Yes, so Jade was working on the spiked micropollutants, basically 5 selected micropollutants, and then she demonstrated the removals of that 5 selected micropollutants. But there are a lot of more various micropollutants in urine. So I'd like to see how many types of different micropollutants, and then how it can be removed by PAC addition.
So you're only working on PAC itself. You're not going to find another adsorbent for this purpose, yeah? Only PAC. And you're going to optimize. All right. I guess overall, with the progress that you have done, I guess I'm happy to see the progress, and also the plan looks like quite clear to me. Of course, it all really depends on how you navigate and try to put your timeline is you still have to work on pilot scale at the end, right? That's one of the plans. So you need to really properly how to say schedule and organize your study if you're going to work on that at the end, so that you can have the time to fully finish with your pilot scale study. Okay,
Yes.
that's all for me. Thanks very much, Gayatri.
Thank you, Dr. Leonard. Weonjung, I just had one question to ask. When it comes to using MBR for urine treatment, has this been carried out before? Has there been prior studies on this?
This urine treatment by MBR hasn't widely done it, and our group started as this lab scale and pilot scale because urine is high strength ammonia wastewater. So, and also in the past, the urine was combined in the wastewater. So using the source separated urine MBR research is quite new to our group.
Okay, so I guess that is the main novelty of your study.
Okay.
No, I'm asking you, is that the
Yeah.
main novelty of your study?
Yeah.
Yeah, okay. And secondly, would you consider the bacteria, the Comomax, as your second novelty of your study?
Yes.
Okay, all right. I do have that concern that Dr. Leonard has mentioned that, if microorganism and microbial is one of the main part of the study, then you need to put in more detailed assessment with regards to that, into understanding the overall performance of the MBR before you can translate that into a pilot scale system. And that would be my comment.
Yes.
Yes, thank you. Now I would go to the co-supervisor, Dr. Sharif. Sharu?
Yes.
We can't hear you. Right, okay, you're here.
Okay. Thank you, Gayatri. Yeah, I think Weonjung did excellent presentation. It's very good way to present. Sometimes I feel like you were reading, but be careful when you some of the sentences are quite long in the slides.
Yeah.
But the way you presented was very clear and very slow and steady. So that was good to me. But yeah, be careful when you are reading from the slides. Not my question, but just one question. I'm a bit confused. Can you go to one of the slides back to where you started presenting on the results and discussion? I'm just okay here when you said that just go back one more. No, okay. This one. In one of this, I don't know where it, but you said that I think maybe I understood wrong way, but I heard you saying that the nitrification rate increased because of the use of this what's the biofilm carrier, right? And then you said that the nitrification rate increased as a result of decrease in HRT. So did you say something like that? Because I was thinking.
Daily nitrification rate is because it's affected by the HRT, because HRT is controlled by dosing pump, and also, so the more it goes and more nitrogen loading rate per day, it will get more nitrification rate.
So when you decrease HRT, that means you are keeping less time inside the reactor, right? So,
Yes.
so I don't see that as a reason for your increased nitrification, because when you decrease the HRT, I don't believe it is. I think it's the way you put into the sentence. I don't think the increase in nitrification is because of the decrease in HRT. I don't agree. I don't think I agree with that. You should. I think maybe it's because of biofilm increase in the surface area of the biofilm, and the more, because when you have more increased area, then you have more nitrification bacteria that is acting, and then probably that increases your and when
Yeah.
you increase, then you decrease, when you increase your nitrification rate, that decreases your HRT. Okay, because
Yes.
HRT is your HRT is controlled based on your nitrification rate,
Yes.
right? It's not the other way around. So
Yeah.
you have to be careful when you say that. So that's one question. So other question is, so have you considered, because when you use the bioreactor membrane and together with this kind of moving biofilm, so it can damage your membrane, because in the long term. So have you thought about that?
Yeah. As I just replied to Leonard, so yeah, in the long term operation, it should be studied. That's one objective as well, for the membrane effect of membrane fouling or membrane abrasion damage. So that's also going to be studied in the long-term study, maybe in the future.
Okay. Yeah, I didn't know because you already answered. Sorry, I might have missed the answer. Okay. think that's all. I think overall you did an excellent job. I think Sean may have some suggestions. Yeah, thank you, Gayatri.
Thank you, Dr. Sharuf. Professor Sean?
Yeah, just answer Yang's question regarding diluted urine. Why we cannot use diluted urine? Because of two factors. One is the economy, and then second one is the environmental impact. Economy impact is if we use waterless toilet to collect urine, so you're gonna have to use your fresh water, which means you can save almost 5 times of your water. So most of people go to toilet every day almost 5 times. So they produce 1.2 liter of the urine. So 5 times. So you will collect 6 liter of. You can save 6 liter of fresh water. So economically, that's a lot of water we can save. So that's the reason why we want to make waterless source separation unit. And second, environmental impact is about, so if we use diluted urine, so you need to wait for your hydrolysis, because our urine is contained as a urea, so we need to quickly hydrolyze the urine to convert to ammonia, and then it goes to the MBL, and then nitrification, and then you collect your fertilizer. That's the one thing you're trying to do. But if you dilute your urine, so your hydrolysis will take a long time, and then it's very difficult to create those more water tanks you have to store. So we need almost 1 cubic meter of the water production from our university every day. So you need 5 times of the storing tank at the basement, so that you need also wait for the settlement of the strobine. After hydrolysis, you create the crystal, and then you need to separate, otherwise this will create your membrane fouling. So there are a lot of implications why we cannot use. We can use, but economically and environmentally, it's much more problematic than waterless. So that's the reason why the Weonjung is trying to use our waterless toilet pure urine as much as we can because easy to handle. Also economically we can convert to fertilizer. That's the answer. And Weonjung, yeah, you need to focus on more about your HLT or nitrification because you are producing fertilizer. So fully nitrified reactor or your fertilizer, it doesn't mean this is good fertilizer because you are producing little bit low quality of fertilizer. So that as long as we can use fertilizer without any full nitrification, so even nitrite concentration is high, as long as it can be used as a fertilizer. Even you don't need a full nitrification. So if you can consider enough is enough. So what's the HLT? As short as possible, and then okay, you don't need 50% of conversion rate. So what's the use of the fertilizer you can produce? It's a Carnomax or nitrification bacteria, AOB, NOB. It doesn't really matter for you. Better is how you can collect fertilizer as cheap as possible, as quick as possible. That's your target, in fact, because you are not producing pure fertilizer. Enough is enough, so it can be used as a most cost-effective way of the production. So, okay, in terms of your PhD, you focus on so many Carnomax or biocarriers and then PhD. Okay, that's fine. That's the good for your PhD and understanding. But in terms of scale-up or commercialization of your research as a PhD, so, okay, no matter what you will produce fertilizer, so how we can make it simple? Okay, we don't care about Carnomax, AOB, NOB, just to produce, and as long as it can be used as a fertilizer, that's fine. Even though there are a lot of ammonia, okay, ammonia is fertilizer, as long as it can be used as without smell, without any kind of some nasty way of the filling. So that's fine. You can use because you will remove most of pathogen and then virus from the membrane bioreactor. So that's also one of your objectives. So in terms of commercial perspectives, why we have to fully nitrify your reactor? Just half or even 5%, 10%, as long as people feel, oh, this is fertilizer, okay, that's fine. So enough fertilizer production, how you can optimize, that is also very important because this is a lot of impact of the scalar issue for you.
Yes.
So that's also think about your research purpose and commercialization purpose. So that's important because no matter what, if you don't consider commercialization, because this research is more about commercialization related research. Yeah. So you have to think about this part more than too much fundamental about Carnomax or AOB, NOB or nitrification, conversion. That's maybe secondary when you look at the urine and the system.
Yes. Okay.
Yeah.
I'll keep in mind.
Thank you.
Thank you, Professor Sean. The panels have all given the comments, so now I am opening for further comments from any of the audience and participants. If there are any, you can unmute and Umakantan has a comment saying great presentation to you.
Thank you.
Okay, I guess there are no other comments. So in that case, I ask all the participants to kindly leave the Zoom session. Weonjung will put you on, don't leave, we'll put you on the in the waiting room and we'll bring you back shortly while the